My name is Rutvik Bedibola, and I will be explaining protein synthesis to you. Firstly comes transcription of the mRNA. In order to finish this, we will need to complete a few steps. We start out with the DNA strand as shown. Red is thymine, green is adenine, brown is cytosine, and blue is guanine. Once we have each side of the DNA strand, we will need to unzip them to copy the codons onto an mRNA sequence. Helicase then comes in and unzips part of the DNA strand that needs to be unzipped and copied for. Once that is finished, an RNA polymerase comes in and attaches itself to the DNA fragment that needs to be copied. In this process, the RNA base pairs come to attach itself to the DNA that is being read. Note that thymine is translating to uracil on the RNA. Once the mRNA is completely copied, the RNA polymerase leaves and the DNA zips back up. Then the mRNA leaves the nucleus to the cell and begins translation in the cytoplasm of the cell. Once in the cytoplasm, the mRNA sequence will get to a ribosome. The ribosome, which is composed of rRNA, will act as a machine to process the mRNA along with the tRNA. The tRNA will be carrying the amino acid, which will be the building block of a protein. The tRNA will be able to connect to the mRNA by anticodons. Now the real process starts. The tRNA now connects to the mRNA, and the amino acid chain begins with the start amino acid. As the process continues, the amino acid chain begins, and the tRNA being used up leaves and gets recycled. Once this entire process is finished, RNA is also recycled for its parts as well. This is only an example, but it continues until the protein is finished and the stop codon is used. That's all for this video, and thanks for watching.